name suggests, soil transmitted helminths are a group of parasitic worms, or helminths, that are transmitted through soil that's been contaminated with their eggs. These infections are very common. It mainly affects poor and disadvantaged communities around the world. It's estimated that over 1.5 billion people in the world are infected with these worms. That's almost a quarter of the world's population. Most of these infections occur in tropical and subtropical countries. They're commonly seen in areas where there's warm and moist climate and there's poor sanitation and hygiene. Although there are many different species of helminths, we'll focus on the three main types that cause infections in humans. These are roundworms, Ascaris lumbricoides, whipworms, Trichuria trichuria, and hookworms, Decatur americanus and Ancylostoma duodenale. They vary in the type of illness they cause and their life cycle. Soil-transmitted helminths are transmitted by eggs that are passed in the feces of people who are infected. The soil can be contaminated by feces in areas where there's poor sanitation and people defecate outside, or when human feces is used as fertilizer. Once they're in the soil, roundworm and whipworm eggs mature into an infective form. This can take a few weeks. Hookworm eggs, on the other hand, mature and hatch into larvae that have the ability to penetrate skin. A person can be infected in several different ways. It can enter through the mouth. This can happen when food such as vegetables are contaminated with eggs and are not cooked or washed before eating, when water is contaminated, when people's hands are contaminated by the soil and not washed before eating. Hookworm larvae have the ability to penetrate the skin of people. People usually get infected when they're walking barefoot. Very rarely some types of hookworm larvae can get into the body when they're consumed. Now, once they enter the body, they eventually make their way to the small or large intestine, where they live and grow. But they have slightly different ways of getting there. Hookworm larvae that penetrate the skin are carried through the blood into the heart and lungs and eventually make their way to the small intestine. Roundworm eggs that are swallowed hatch into larvae, enter the blood, make a trip via the heart and lungs, and eventually make their way to the small intestine. Whipworm eggs, on the other hand, hatch into larvae and make their way directly to the large intestine. Once in their final destinations, they attach to the wall of the intestine, grow, mature, and can live there for many years. They also produce thousands of eggs per day, which are then passed out in the feces to start the cycle of infection again. People can be infected with more than one type of worm at the same time.
Sample preparation. Place an empty beaker on the scale and press the tare key. Add 2 grams of the sample to the beaker. It is important to weigh exactly 2 grams as this may influence the accurate estimation of the fecal egg count. 30 milliliters of the flotation solution is added to the sample. Homogenize the sample using the spatula and pour the fecal suspension 3 times through a tea strainer to withhold the large debris. To obtain optimal results, it is crucial that the debris is firmly squeezed after the sample is sieved for the 3rd time. Once the debris is squeezed to dry, it can be discarded. Filling the slide will be simplified when it is rinsed and tapped on the surfaces. Before filling the slide, be sure that the external surfaces are clean and dry. Place the slide on a flat surface ready for filling. Homogenize the filtrate by pouring it 10 times from one beaker to another. Immediately place the tip of the pipette in one corner of the chamber and squeeze the pipette until the chamber is full. Do not move the pipette across the opening of the chamber as this may introduce air into the slide. Re-homogenize the filtrate and repeat the procedure for the 2nd chamber. Examination of the slide.
Press the tare key and add exactly 2 grams of stool with a wooden tongue depressor. Afterwards, add 38 milliliters of the flotation solution with the correct specific density. After closing the fill flow tack, remove the cap of the applicator so that you can thoroughly mix the suspension by moving the applicator up and down. In order to fill the mini flow tack, it is recommended that you tilt the mini flow tack using a 1 cm thick rectangular prism. Make sure that the slits are on top of the chambers. Take the fill flow tack and remove the cap of the other opening so that a pipette tip can be screwed on. It is recommended that you cut off the top of the tip to avoid its obstruction while filling the mini flow tack. It is essential that the suspension is homogenized again just before you fill each chamber. Fill each chamber by squeezing the fill flow tack. To avoid air bubbles from the remaining procedure, the chambers should be filled until the meniscus is formed. After an incubation period of 10 minutes at room temperature, use the key to turn the disc. The grids are now perpendicular to the chambers. You can remove the key but keep the base and the disc together. These 2 parts should be placed in the microscope holder with the chambers sliding in the gap in the middle of the holder. Place the microscope holder and the mini flow tack on the microscope table.